And thanks, Mary, for saving the youngest till last. So I've obviously got the shortest career of all the speakers, so hopefully I have something for everyone. But this is going to be a bit different to everyone else's because I want you to ask me questions. There's not much point in me giving you stuff that I've done over 30 years and assume that that's what you want to hear. I want you to ask me questions as I'm going along, okay? So essentially, I'm going to give you a snapshot of 30 years working in different aspects of what can happen after the course you're in or going through. So this is what happened from the 80s to 2001. So I started off, admittedly, it's a confession. I didn't know it was only for graduates at GMIT when I was invited. But could say nothing, because I've actually living down here for 25 years anyway, so it shouldn't be a problem. I'll sneak out anyway. But I went to a parallel universe, essentially, where we did the same kind of course setup in Dublin as was done here in the West. So I started in DIT Kevin Street, the equivalent at GMIT. Now, initially, it used to be a cert course, 3-year cert course, 2 years in college, and then 1 year actually on the job, in a training environment similar to your 4th year. And my training was in James's, so I got a broad range of lots of different techniques. A very large hospital, similar to GUH, and it set me up basically for anything I wanted to do afterwards. Then after that, I went to Piedmont Hospital. It was the start of 15 years in the lab, so I spent half my career doing lab work, half it doing public health work. And Piedmont Hospital was quite a small hospital on the edge of Dublin. And a lot of people told me, don't actually go there, it's a bad career move as your first job. But I chose to ignore it, which a lot of people here seem to be ignoring advice as well, so why not? But I'm glad I did, to be honest with you, I'm glad I went. Now, admittedly, the advice was don't necessarily do it as a long-term career move, but it was portrayed in a way that, well, maybe you shouldn't do it at all. Maybe stay where you are, stay in the one place you were trained in. But I had a fear that if I stayed in the place I trained in and kept on going up the ranks, I'd always be viewed as a student who stayed on. So I wanted to go away and actually do something somewhere else, get a bit more experience. So because this was a small hospital, it was multidisciplinary. So I had to rotate between the different departments, which is quite useful. Because when you move to certain hospitals, if you're on call, you may actually have to cover blood bank or hematology or biochemistry all at the same time as your own discipline or your own preferred subject, sorry. So that was just about a year, and the only reason I left is because I had to. They were bringing in cutbacks, they were going to let one person go, and last in, first out is usually the way it goes. It was either going to be me or someone else who had a family and a mortgage, so I chose to go. So I went back to college and did the diploma, which was essentially a 1 year course that used to be split into 2 6-month blocks with a year's in service in between. But because there was a shortage of jobs, they had to rearrange the course, and they allowed you to do the 2 6-month blocks back to back. So everything was out of the way in one sweep. So that was quite handy. So after that, I changed tack in my career again, and I went to ICP in Dublin, which doesn't exist anymore, but it's nothing to do with me. I left long after it went down the tubes. But that was actually a clinical trials place, and I always was interested in micro. And what I ended up doing in the diploma was specialising in micro. I presume you've read it ahead, even though I didn't mention it a minute ago. And I used to do micro for phase 2 and 3 clinical drug trials. Basically, they were testing new antibiotics. I had to do the usual kind of sensitivity tests, using the same kind of organisms and so forth, and using FDA type regulation, as one of the other speakers mentioned. And this was a precursor to accreditation. A lot of places didn't use these kind of rigid, standardized techniques, but we were FDA approved as well. It was an unusual experience, but one of the advantages of actually moving sideways like that is I saw the non-health side of things. This was like a private company. It wasn't in the health service, and it was still a relevant discipline, very similar work, but it actually exposed me to the LabStar computer system, which is an older system by today's standards, but that actually landed me the job in Galway further on. So the reason I'm even mentioning that is sometimes a sideways move may seem like a daft move or a bit risky, but actually some of the things you pick up along the way will actually help you out further on, and it may not always be apparent what direction you're going in at the start. So after that, I actually went back to James's full-time employment, because I was worried that I wouldn't actually see enough of the stuff that I'd need to see to get through my exams, going forward. So I thought, no, I need to get out of the research side of things. And I went back, got lots of experience in very specialised areas in James's. And again, that actually helped me go further. I then was faced with a dilemma because I had a 6-month contract being offered. I was on a succession of short-term contracts, maybe a month here, 6 months there, or whatever. Eventually you get cheesed off. As you move on in life, you actually want to have an anchor, somewhere to actually put down your roots or whatever. And I thought, well, this is getting a bit much. Maybe I should look elsewhere. And I also was afraid of being pigeonholed as being the student who stayed on again. So I thought I'd chance my arm with this. I had a friend who actually mentioned that there was a possible job going in GUH, and I just chanced the phone call, was talking to the chief, and everything was hunky-dory. He seemed to actually want the experience I had. But the clincher was the previous computer experience. It was the very system they were implementing. That's why they were recruiting. So as I say, sometimes a sideways move has benefits further on. So that brought me to Galway. So I was there for 10 years, so I would have met some of your predecessors, students going through, and to be honest with you, they've always been brilliant. And I'm not just saying that because I'm here, in case I don't get out of the car park. And similar to the presentations that have gone ahead, very high quality of students, really, really knowledgeable, but still always ask really good questions when they're in the lab. So while I was in GUH, I went on again. I went and did a MSc in Medical Micro in Bristol. And the reason I did that was there weren't too many options for MScs here. There was Coleraine as one option, which some people went through, but I actually preferred to go to Bristol, partly because my folks are in Dublin. I could fly from Dublin and see them on the way home or on the way out. So stupid idea, but I mean, it actually worked out well for me anyway. So after that, I got sick of the lab, basically, as some of my previous speakers have actually done in a shorter time period, I might point out. And that's actually why I asked yourself, why did you miss the lab work? I'd actually got to a point after 15 years where I didn't really think I was going to see anything more interesting. Like at a certain point in micro, you'll see so many of the unusual bugs that you think, well, what am I going to see after this that's going to be new? Now, admittedly, every job becomes routine after a while, so that would creep in anywhere. But I'd seen this type of job advertised in around 1999. Now, there's a national surveillance centre. It's now called the Health Protection Surveillance Centre, based in Dublin. You may or may not have heard about it. I believe a little birdie told me earlier that people were being told about notifiable diseases, so I might ask questions in a minute and see if you were listening. Not to me, but to your lecturer. But in around 1998, HPSC was set up as the National Disease Surveillance Centre with those initials. I don't know why they rebranded, but this happens a lot, same with the health boards becoming the HSE. There's no identifiable change in what they're actually doing, but they decided a name change actually makes things better. So they were started around 1998, started recruiting surveillance people in 1999. They started off with 2, and the idea at the start was that they'd use people who came up through the medical laboratory science, as it was called at the time, or medical science now, that pathway, with a special interest primarily in microbiology. Now, not exclusively that, because you would have done some of it, leading up to your degree level, so you'd probably get in anyway. But there was a divergence away from the original plan, so now there's a mixture of people from various different backgrounds. At this stage, I'd say there's 3 out of maybe 11 or 12 public health-based surveillance scientists who have my kind of background, have come up through the lab system. Whether they did PhDs, MScs, or whatever is irrelevant, but they all came up through a similar kind of pathway. But a lot of the other graduates would have done other science degrees or had research experience, in the likes of Chagas, as someone else mentioned. What did I want to say after that? You'll have to forgive me, I'm a bit older, so I'm inclined to forget what I'm actually going to talk about next. I'll just have a look at my cog sheet here, which all people going to exams should have with them. Oh yeah. So essentially there's 1 or more surveillance scientists in each public health department. Now, each of the old health board areas is essentially a HSE area. Now, again, it's back to rebranding. It didn't change anything. We still do the same job, work in the same areas. And it's not standardized the way things are done. As in, people are recruited on different salary scales. They come from different backgrounds, so they don't share the same kind of knowledge, or background knowledge, which in some ways is good for the likes of me, because I understand a lot more of this kind of stuff. I came from that background, so I understand what the lab results mean, whereas people from a pure science thing, they have major headaches trying to get their head around the lab results and what they actually mean. So now it's your turn. So it's audience participation time. Who knows what a notifiable infectious disease is? Don't be shy. I might ask for a volunteer. Or a point. Okay, I'll make it easy for you. a notifiable infectious disease is one of a scheduled list of diseases that are obliged to be notified to a medical officer of health, is the actual term. And basically any clinician, I'm going to answer the 3 bits all in the one go. Any clinician who actually diagnoses one of these listed diseases is obliged to notify the medical officer of health. And that person is actually a doctor. Medical officer of health is a function held by either director of public health or his nominee, and they can only nominate down one level. So it'll be a specialist in medicine or the director. There's one of those per health service area. So you can do the maths. There's less than 12 in the country. So who notifies? Any clinician who diagnoses one of those conditions. Now that's a GP, hospital consultant, or both. And there's also an obligation on a clinical lab director. And essentially it's usually not the actual director of the lab themselves that notifies, it's a nominee. And that typically is a lab-based surveillance scientist. So there's 2 aspects of this job. Mine is public health based. I work in a non-lab environment, which our friend over here who didn't like actually lab work would probably appreciate. It's a desk job. And my counterpart in the laboratory does both sides. So she feeds information over towards me. I willingly accept it and I don't have to get my hands dirty in the lab, which is kind of handy in some ways. Why do people notify? Well, A, it's a legal obligation. So then you have to ask, well, why is there a legal obligation for these things to be notified? A lot of diseases, when you make a diagnosis, you're actually addressing the patient. You're actually finding out what's wrong with them, can we treat them, and how best to proceed. With some diseases, as in the ones listed, there are actually ramifications for the general public as well. Let's say someone is diagnosed with Ebola and no one thinks to tell anyone else. What do you think happens? We wouldn't all be here today. Half of us would be gone. And that's the most extreme version. It hit the papers relatively recently and it's very much in people's mindset. They think that's the killer bug, that's the big problem. But even things as innocuously sounding as mumps or measles, they have ramifications as well. People can die of either of those. And that's apart from other complications like brain injuries, orchitis, which is very unpleasant if you're a male. It involves swelling of the gonads. I won't go any further with that. And it can lead to sterility. And measles is incredibly infectious as well. Part of the reason it's on the list. If I walk into a creche as a toddler and I have measles, and there are 10 people in that room with me, 9 of those people are going to go away with measles and go on to infect and infect and infect. And before the introduction of the measles vaccine, it was a killer. In the year 2000, because of a fall off in vaccination rates after a paper that was published in 1998 by Andrew Wakefield, there was a lack of interest, well, it's the wrong phrase, there was actually a lack of trust in the MMR vaccine, and it was completely biased. It wasn't a very honest paper. It should never have been accepted for publication, but it did huge damage. And in the year 2000, 2 years after that publication, because of the public outcry and the lack of trust of the vaccine, 2 kids died in an outbreak in Dublin. There was about 1,800 people involved, hundreds of those hospitalised. I don't even know how many ended up with more serious complications like SSPE, subacute sclerosing panencephalitis is one of the complications. Pretty nasty. It doesn't happen that frequently, but it's a very unpleasant complication. So some of the diseases we tend to roll off the tongue, like measles and mumps being, you get swollen cheeks, with, chipmunk features with the mumps. That's a fair description for most people, but it isn't for everyone. And it was the most common cause of viral meningitis until the introduction of the MMR vaccine. It's rarely a cause of viral meningitis now. We've had 1 or 2 in the last 5 years. It's so rare. And we've had massive outbreaks of mumps in the last few years. So it just goes to show the effect of vaccination on a decrease in disease rates. So that more or less covers the why notify. But the other aspect is that some of these diseases like Ebola, the more epidemic proportion diseases, smallpox, anthrax, the bioterror kind of agents, they will always be on a list of notifiable diseases in every country that's a signatory to the WHO because they have such global significance. And we have to make immediate notifications to WHO of certain diseases. Like cholera be one, obviously Ebola be another one, smallpox, polio, those kind of ones. It requires immediate action because it can sweep across the planet very quickly. Oh, sorry, forgot to press the button. So what is this? I don't know if that's in focus for everyone out there, I can barely see it from here. But it's actually the list of notifiable diseases. And we're going to spend about 2 hours now going through each and every one of them in detail. I'm sure you'll be glad to know that. Basically, I'll shortcut it in case anyone wants to count them. There's 83 listed. Some of them you will see zeros in, they won't actually be reported, but we still have to be aware of their presence. The likes of anthrax will be up in the top left. Most recent addition is on the bottom right in Zika. No surprise there because it's of global concern and interest. And we also have the more exotic ones, the smallpox, and Ebola is in amongst the viral hemorrhagic fevers there towards the bottom. So that's the full list. Most of those we actually get cases of, but there's a few that hopefully we'll never see cases of. But just because we won't, it doesn't mean we shouldn't be prepared. So people actually are always producing protocols to actually deal with and contain and share information on potential cases of Ebola, potential cases of smallpox and so forth. So there's a readiness there, it has to be there. We have an isolation facility based in the Mater Hospital for people with Ebola. You will have seen very unfortunate pictures maybe during the Ebola, I was going to say pandemic, the Ebola scare in West Africa, where an exercise in Dublin was actually being photographed, and there were some very unfortunate pictures in it showing things actually not going too well. You had a guard with no protective gear on standing beside a guy in full bodysuit, which didn't look right to me or any of my colleagues, but actually probably looked fairly okay to someone else. It was kind of a scary picture to have, but it was an exercise. It wasn't actually the end of the world anyway. What else would we have there? Nothing else. Let's skip on to the next one. And you can ask questions if you want. I don't mind you interrupt me as we're going along. I'd rather you do that because I appreciate this 30 years of stuff here I'm trying to cram into a few minutes, and I don't really know what to focus on because the job I do now is relatively rare in terms of there's only about 12 of us doing it in the country. There may be more opportunities coming up anyway, and I'd rather you ask me whatever you want to know rather than me telling you what I think you might like to know. So what is my role? So I'm basically involved in the surveillance of notifiable infectious diseases for our 3 counties in HSE West. Now what's in brackets there is CIDR, and that stands for Computerised Infectious Disease Register. We use a computerised system for actually notifying our diseases. Now the legislation actually doesn't specify we use this. It actually specifies something with a slower turnaround time. It actually goes back to the old days of paper notifications, where a clinician would send in a paper slip into their public health department, which is where I work. And every week we would have an obligation to make returns to the national body, formerly NDSC, now HPSC. And that's a very slow system, as you can imagine. For something like Ebola, you don't want to be waiting for the postman to deliver something so you know what the figures are down the country, because half the country will be gone by the time you actually find out what's going on. So in 2004 they piloted this system. It was custom built in Ireland, or not in Ireland, for our very specific set of parameters. We wanted to tailor it to the diseases we had. We wanted to be able to add extra information, like your core notification is essentially like a credit card slip, essentially if you look at the paper version. It's got the name and address of the patient, date of birth, where they might live. I'm sorry, that's the address obviously. Clinician, what the disease is, crucially enough. Sometimes when the paper ones come in, the doctor has forgotten to put what disease the person has, which is not very helpful, so they usually get a phone call back. The date of onset, and various basic things like that. And there's a subset of diseases on that list where we are required to gather extra information for our National Surveillance Centre. And things like cryptosporidium, we'd have to gather information on maybe the water supply, what risk factors they may have. Do they live on a farm? Have they visited an open farm, pet farm, or like that in the recent past where they might have picked up the infection? Whether they've been to a swimming pool. Some of these are related to whether they've actually acquired it somewhere or whether they may pass it on somewhere. We would also be asking details about their parents' occupations. Are they healthcare workers, where they could pass it on to someone who might have a compromised immune system? Do they work in the food industry, where they could pass it on to someone on their plate if they're not washing their hands or whatever? So there's a lot of extra stuff we gather on certain diseases. That's a small subset of what we gather information on. The TB data is humongous, like there's 72 questions I think on the TB form, so I'm not even going to start listing those. Crypto was the one that sprang to mind, so I thought I'd start with that one. So what do we do when we talk about surveillance? I'm basically looking for trends in infectious diseases or trends in notifications. So you could start with bare numbers and look, have we got an increase in this disease versus that disease for the same period last year or over the successive years before that? And it can be very simple. It can be based on memory, as in you might remember getting the case of such and such a disease last week from the same area. You think, well, potential outbreak here, whatever. And that's essentially what I'm supposed to be doing. I'm supposed to flag potential outbreaks or potential problems and take it to the medical staff. And we decide then whether there's a need to create an outbreak or have an outbreak meeting to discuss further, get more test results. Not necessarily test results on humans. It could be environmental test results, like water test results and so forth. So that's the way it cascades. You start off monitoring, then you go on to flag things to the people who actually would be taking the action. And it'll probably make more sense when I get onto a few slides that I have for you that will actually show the various things that would have been investigated. So I would serve as a member of an outbreak control team. Essentially, that's a multidisciplinary team meeting which would involve medical staff within the public health department, the likes of myself. They would often be consultant microbiologists. And depending on the disease, you may have veterinary people involved, you might have the county council. A lot of the time we have meetings with Irish Water or the relevant county council because there's often a water component. Things like crypto would be a great example of that. And we'd add on extra members depending on what was actually going on. For things that might cause panic, we may actually have the emergency services involved, the guards may be involved. There could be public order implications of making an announcement about certain diseases. So there's a lot of disaster planning goes into this as well. It's not just a react straight away. There can be multiple different agencies involved and it has to be cascaded properly. And everything is confidential. If there was a leak of something that had potential to cause panic in the public, that would not be in anyone's interest. So everything is kept mum until it's released in a controlled fashion. That's why every time there's an outbreak, you get a statement released. One. We've had incidents in the past where there have been multiple little leaks, mini statements, for whatever political reasons, and they don't help anyone. It causes confusion because they often have conflicting information in them. Anyway, that's more about the workings of the public health department rather than my own role, so apologies for digressing. So what would my role be on the outbreak control team? So it's the next thing. It's essentially data analysis. We often have cases where I will bring something to the team and I would show them the information I've gathered, like the trends in such and such a disease. I might have maybe seen an increase in cases over a period of weeks in a particular area, in a particular age group, gender, what else? Ethnicity on rare occasions. I'll take it on and we'll see if that needs to be done. It can also go in a different direction where we get a call from Irish Water, say, and their routine monitoring of the water supplies has revealed crypto in the water. So this is in the water as opposed to in a human. They would often contact me and see, not directly, via the Director of Public Health, to pull out figures for that area over the recent past or over the recent few years and see if there's anything odd happening now versus the past. So I'd have to look at the more recent figures relative to the recent crypto positive in the water and see if it's any higher or any lower than previous years. And it can be the basis for a boil water notice being put on. And I'd say most people here would have been subject to a boil water notice or lived in a boil water notice at some stage in the last few years. They're quite common. And sometimes it's due to treatment failures, sometimes due to a mechanical breach in a pipe. There's always people with JCBs going around digging things up and they never know what's under the ground until they actually stick the bucket into it. So that's part of the data analysis aspect. So one is from first principles where I actually see something odd and I flag it. Another is I'm actually asked for information about something that they suspect might be going on before it may even have gone on. So I would reiterate or confirm that there's a problem. And there's also a data analysis aspect of, let's say, a foodborne outbreak. We would often get, I was going to say complaints, but allegations that people who attended a function, maybe a wedding or a graduation or something similar, hopefully none of you will be affected by a graduation. Well, you know where I am, I'm just in Maryland Park here. We'd be told maybe about 10 people out of 20 who attended a function came down with the same kind of symptoms within X amount of hours out. So I might have to go and do some data analysis on that, maybe based on the actual menu that had been served. How many people had what out of the menu, and if they did have that, how many portions did they have? Did they have water? Did they have wine? Whatever. And we try and work out the most likely food item based on risk ratios. Very simple calculations, not high-end maths at all. So that would be 3 minor things that I would do in terms of data analysis for outbreak investigations. I'd also be a lab liaison person. Like, I would link with my counterpart in the laboratory here in Galway. If anyone's been trained in Galway in micro, you might have met Belinda. And there's also another person up in Mayo, Magella, and there's another one in Portiuncula, Michelle. So they'd be the 3 local labs that I'd be linking with. But I would also link with reference labs. We have a Salmonella reference lab here in Galway. There's a Cherry Orchard reference lab for VTEC, toxigenic E. coli. We have a TB reference lab in James's. The Meningo reference lab is in Children's University Hospital in Temple Street. And a lot of that will be to determine whether or not we do or don't have an outbreak. Like, having X amount of cases doesn't mean there's an outbreak of that disease. You also need to see, is it the exact same type of the organism? Like, if you have 5 salmonellas in the space of a week from a small town, it probably is an outbreak. But if they all turn out to be different types of salmonella, then it more than likely isn't. It could be coincidence. Maybe they're on holidays in Spain or Portugal, wherever, all came back with a dose of the you-know-whats. And then the final part that I put down here was liaison with HPSC. They're the national body for surveillance. They would often come to me for clarification on various things we'd actually have notified. At the bottom of the list of notifiable diseases I gave you, there's actually a little thing that you probably wouldn't have been able to see. It says refer to case definitions for further. Each of the diseases has different categories for notification. You could have a confirmed case, probable case, or possible case. There are slight variations. Sometimes there is no possible case or probable case. And they usually follow the format of possible cases. When a doctor diagnoses something or thinks that someone has something, that's a possible notification, or a possible case, and is a clinical notification. We also have a confirmed case, which is a clinical case that has been confirmed by a lab test result. Where my role would be quite useful in that is I also would know, well, does that result correlate with a confirmation, a true confirmation? Because if you have someone who, let's say, had been vaccinated with MMR, and they're tested for measles, and they have IgM positive to measles, well, that could indicate either active infection or prior vaccination with MMR. So that wouldn't necessarily be conclusive. So I would be looking more for a molecular result, where you actually look for the genetic material of the living organism rather than a result that might indicate past exposure or past immunization. So that's probably enough on that anyway, before I go waffling on anymore. I tend to sidetrack myself. So this is an example of the kind of thing that I'd be doing. So I'd be up all night now with the crayons drawing this kind of rubbish. And this essentially came from a dump from the national computer system of a particular disease. I'll tell you what the disease is in a minute. And I basically take it into Excel. And on the bottom, you see all these numbers, 1 to 52, just in case you can't see what's written there from the back. That's basically the week numbers in the year. All the coloured bars at the bottom represent the different years. So this is one disease over the course of the weeks of the year, by year, and the number on the vertical column is the actual number of cases we had in that year. What I'd like to point out first is, from here around week 13 here, over to week 26, that's essentially quarter 2 of the year, which is spring. Sorry, my eyes are getting a bit sore. So that's spring, and what I want to show you, ignore the big blue peak for the moment. There's a clustering there. You can see there's definitely a hump in virtually all of the actual traces going across there during spring. And that's normal, essentially. And that's what I'm supposed to do, is see what's normal versus what's abnormal. And this is a very simplistic view of it. And it also is intended to show you, well, that thing sticking up there is obviously abnormal. Okay? But what my job is I have to see that before that happens. And if I tell you that that was Cryptosporidium, would that surprise you? That was 2007. Okay, so a lot of you would have been living in here or visited here or had to stop drinking the water here because of that happening. And actually, what happened with this is, you'll see here, it starts over here, it may not be clear from the back. There's a little hump there, which is a bit unusual. And that essentially was little clusters of cases out in outlying rural areas. A little bit earlier than normal. Not that abnormal though, because crypto can be got from animals. It's one of these things called a zoonosis. It's not only from animals, because there's 2 different strains that actually predominate. One is hominis and one is parvum. Parvum can be in humans or animals, so that's the one you'd most likely get from the animals. But hominis is not carried by animals. And this outbreak was caused by a mixture of the 2, mainly hominis. So basically, when we were getting these ones here, we didn't know what type they were anyway. But because they were in rural areas and there didn't seem to be any obvious links to a common factor. They all lived on farms or close to farms. It was assumed that this was related to that, and it probably was, because that's what most of these ones here in the spring peak are. It's to do with calving and lambing. Young animals more susceptible to the infection. They excrete it in large numbers in their feces. Ends up in the water systems or on people's Wellington boots, into farmhouses, 2-year-old crawling around the ground, fingers in the mouth. Classic. Well, not so classic for the kid though. But it can actually leave them very debilitated. I mean, it's an illness that can actually take 2 weeks to clear, and it's quite unpleasant, as anyone who actually got it would know. But this peak here was actually spotted in around week 11. We had boil water notice on in around week 11. So it was this segment going up that was noticed that we were getting cases that weren't just in the rural areas. Sorry, I left my teeth at home. My teeth tripped me up there now, chewing them. We noticed these people weren't all from rural areas. Maybe I should say they live in the country. Easier. We were getting a lot of urban area people who had no contact with animals, no other risk factors. They hadn't been to swimming pools. Initially, with some of these initial cases down here, we had actually noticed that they'd been in swimming pools as well. So we thought, well, it could be one person who has a fecal accident in a pool, contaminates the pool, and it's actually surprisingly common. Some people actually do it deliberately on holidays. Get a free holiday. Yeah, good, you know, it's called logging, apparently. Anyway, I digress. Sorry, I've distracted myself again. No, definitely not. No, it's nothing to do with me. Meanwhile, back at the chart. At the bottom end of that leg there that's going up, we did notice some people had been to swimming pools, but they were never the same swimming pool, which doesn't prove or disprove anything. It's just an additional risk factor. But by the time we reached that little shoulder there, it was starting to be more obvious that we were getting cases with no other obvious risk factors or exposures. At that point, we were getting worried. So we had our outbreak meeting, we had environmental health involved as well, and we were in with the council. And it was decided in the interest of public health, public safety, we should consider a boil water notice, even as a precautionary measure. So this is around week 11 going into week 12. It's just before Paddy's Day in 2007. 2007. We're going all Michael Noonan, leaving out a zero. But it was another week to 2 weeks before we actually got confirmation that it was in the water. And for those 2 weeks, there was a lot of disbelief that this was actually going on at all. Now, can anyone explain to me why that peak went up when we put on the boil water notice there so early?
Because the cases maybe were just still coming in?
There's a small bit of that, yeah. The actual reason is the incubation period for crypto is about 2 weeks. So we put it in there. So people who might have actually drunk the water for the previous 2 weeks hadn't actually presented with symptoms yet. And that doesn't include people who pass it to their family members after that as well. So that will actually propel the spike upwards. So you can imagine where that spike would be if that boil water notice had gone on later. So we ended up with over 300 cases that year, and normally we'd have anywhere between 80 and 120 cases in a full year. And we had most of them in that period in March. And the boil water notice extended from essentially that shoulder there all the way over to, I think it was week 32. After that, it was all fine. It required a complete changeover of the water treatment plant. It was long overdue. Anyway, that's as much as I want to say about that. But that's probably the most visual example of actually what I do or what I'm trying to do. And that was.
Did you set the threshold yourself, or did someone tell you that the threshold you should be aware of?
Which threshold? Sorry.
Say, for example, when it got to the shoulder, right?
Yeah.
Was it at that point that you knew to react?
No, actually, no. There's continuous monitoring all the way along. It's one of the diseases we do what's called enhanced surveillance on, and we look for people's individual risk factors beyond the core data of their name, age, address, and all that. So by actually looking through all the feedback we got, it was gradually becoming apparent that we were getting people who didn't have any other risk factors other than water. So there's no threshold as such. It was just that little shoulder there kind of corresponded with when we were starting to see an increase that was mainly due to people in urban Galway areas who had no contact with animals. Couldn't possibly have got it anywhere else other than the water, or else there was something major going weird.
What's the peak there around week 15, 16, 17?
Sorry?
See the second peak, smaller peak. Around week 17.
Week 15, 16, 17?
Yeah. Was that boil water still on there? Or was it boil water?
Oh no, that it continued on. The boil water notice was still in place, but you also have to factor in the fact that there'd be people who'd be getting sick going up in that spike, who can infect other members of their household, they'll present later and later. And there's also an increased awareness when there's a boil water notice like that on. People will go for testing more readily. Like, how many people here go to the GP if they get a dose of the trots? You don't tend to go in a hurry. What you tend to see, and what was happening at the early stages, is it's very young children are brought to the GP because there's more concern about a very young person or someone who has an immunocompromised condition, or someone who's much older who will be more susceptible to dehydration. So we were starting to see younger people at the start, and then it was affecting all age groups. So that double peak isn't actually something actually going wrong. Again, it's more it's like a reflection of what went up, but on a smaller scale, and it usually is family transmission. And that's probably only a snapshot of what was going on, because these are confirmed cases. The actual number of people who might have had diarrheal illness is probably way higher. There have been estimates that it's probably 10 times what we actually got. So you've got 2,000 to 3,000 people, which is quite substantial. So 70,000 people essentially were discommoded in terms of their water supply to avoid 70,000 people ending up with that. So that's an example of when it actually is working right. It's quite hard to actually, sorry?
I'm just conscious of the time.
Sorry, apologies.
Sorry.
I will scoot on. I'll scoot on. There's only one or two more.
We'll just,
Yeah, I'm just going to skim through this.
yeah.
Okay. H1N1.
Just don't need to miss the room.
That's fine. No, that's great. I'll wrap up now in a second. H1N1, swine flu. That blue peak there towards, you don't have to read this, that's just a succession of flu isolates over time, going from 04 to 16. That's the pandemic flu. And you can see, because there's a vaccination campaign, you don't see much of that colour further on until you get towards this year. That's the predominant colour this year. We had a few hundred cases this year. People would have got the vaccine and have knocked it out then for quite a few years, and it's starting to come back again. That's all I wanted to show you on that. WHO actually formulated a vaccine for flu based on this kind of stuff. They would see what's circulating in the hemisphere. It's like the flu season is split into two hemispheres in the world. Its monitoring is in the winter period for both halves, and they formulate the vaccine for the next season based on this kind of data. What's the predominant circulating strains? That's all I wanted to show you on that. This one is, what is this? Measles. You can easily spot the increase in cases in the middle, okay? There were a succession of outbreaks in both of those years in the middle. But what I want to show you is it's not just pure numbers and something appearing as a spike. On the extreme right, you'd see a very small bar in 2016. That's actually an outbreak of 2 people. And the only reason we know that is because they took place in a very short space of time with each other. One person came in and infected another person. So that's actually an outbreak over there. So it's not always big spikes in the sky. This, okay. I believe you've been talking about meningococcal vaccine group B to some students, and can anyone explain to me why meningococcal group C is actually so low on this? It's the same format as before, years gone across the bottom, numbers of cases up here, and group C meningo is very, very low there. Okay, I'll short circuit it. Essentially, group C meningo vaccine came in 2000. That's why it's so low. Before 2000, you'd have the same kind of spikes in the group C column as you do in the group B column. And hopefully when the B one comes in, that'll go down to those levels. But what tends to happen is a different group shoots up as the predominant group. But hopefully the numbers will be lower. Again, we'll scoot on. Now, if anyone wants more information, I'd certainly recommend you have a look at the HPSC website. There's lots of stuff on there. Also, in your in-service training year, if you're in any of the larger hospitals, you will probably meet one of the surveillance scientists in the lab. They will be able to tell you what they do, because it differs slightly to what I do. There's links between us, but they're not the same function. The other thing I was asked to do was give advice to my younger self. I wrote it on the page, but I forgot to put it in the slide. Essentially, my path to being here has been very kind of twisty, turny, all over the shop. And I would say don't be afraid to make a choice that actually takes you in a different direction to what you might have originally thought you'd go in. Because it's all experience. You can't lose. And you can always go backwards again or change direction again. And the only other piece of advice I'd say is be good to the students. Remember what it was like when you were going through. I still remember. And I'd like to encourage people the same way as I was encouraged. Some people don't want to engage. But don't be those people. Do try and help your fellow students when they come into it. Assuming you actually want to work in a hospital. And that's me finished. Sorry, Mary.